and welcome to my channel, Sam Biology Class. Friends, today we will discuss about fluorescence in situ hybridization. Before that, will you please consider subscribing my YouTube channel? Your subscription is often a lot to me. Fluorescence in situ hybridization or FISH is a molecular cytogenetic technique that localizes, identifies and visualizes specific DNA sequences on chromosomes in cells. In molecular biology, hybridization means base pairing between complementary nucleic acid sequences. Fluorescence means the emission of light, and in situ means original location, which in this technique is the cells. Because this process involves hybridization between chromosomal DNA sequences and fluorescently labeled probes that emit light, it is known as fluorescence in situ hybridization. FISH has several applications. Number one, it is used for identification of chromosomal aberrations such as deletion, translocation and amplification. It also monitors bone marrow transplantation success. Number two, this technique is useful for detection of chromosomal imbalances such as aneuploids. It monitors genetic diseases such as cystic fibrosis and Down syndrome. In addition, FISH is used for gene mapping and for identification of novel oncogenes. Finally, this technique is used for identification and quantification of unculturable microorganisms in complex environment such as oral microbiome. In fluorescence in situ hybridization, fluorescently labeled probes are used to detect specific DNA sequences on chromosomes in the cell. Probes are oligonucleotides, either DNA or RNA. Because of high degree of complementarity, they base pair perfectly with the target sequence. For FISH, probes are between 200 and 300 nucleotides long. In order to prepare the probe, we must know the nucleotide sequence of the target DNA with which it will base pairs. Probes are prepared either by synthesis or can be isolated from several key sources. In next step, probes are labeled with fluorescent dyes. There are two different methods by which probes can be labeled. One is direct method and the other is indirect method. In direct method, fluorochromes are directly attached to the probe nucleotides. Now the question is, what is fluorochrome? Fluorochromes are fluorescent dyes that are used as direct label. Different types of fluorochromes that are used in FISH are cyanin dyes, Alexa fluor dyes, fluorescein and auto dyes. This figure illustrates the process of direct labeling in which fluorochromes are directly attached to the probe nucleotides by a process known as neat translation involving enzymes deoxyribonuclease and DNA polymerase 1. For example, this is the double-stranded DNA from which the probe has to prepare. First of all, this DNA is treated with deoxyribonuclease, which will remove nucleotides from random location from one strand. These are the nucleotides which have been removed from different locations on one strand. This action creates single-stranded niches which are then filled with complementary fluorescently labeled nucleotides by DNA polymerase 1. Now you can see that in this double-stranded DNA fragment, only one strand is fluorescently labeled while the other strand is not. This is followed by denaturation. By raising temperature to 75 degrees centigrade, these two strands are separated, out of which only the strand which is fluorescently labeled is used as probe to detect specific DNA sequences on chromosomes. This is about direct method of labeling. Second method of labeling is indirect method, in which the probes are first labeled with haptens. Haptens are chemical compounds which themselves are not fluorescent. Two types of haptens that are used in fish are digoxygenin and biotin. Digoxygenin labeled probes are identified by using fluorescently labeled anti-digoxygenin antibodies. On the other hand, biotin labeled probes are identified by using avidin or streptavidin conjugated tags. Out of these two different methods, indirect method is more sensitive. These are the key steps of fluorescence in situ hybridization. First step is sample preparation and fixation, in which the target cells present in the sample, such as blood, bone marrow or tissue, are fixed on a microscope slide using methanol or acetic acid or formalin. Fixation is essential in order to preserve the morphology of the cells. This is the cell which has been fixed on the microscope slide. Second step is pre-treatment, for which the slide containing the target cells is treated with a proteolytic enzyme such as pepsin in order to increase probe accessibility. Treatment with pepsin makes it easier for probes to enter into the target cells. Third step is denaturation, for which the slide is heated to 65 to 75 degrees centigrade. At such a high temperature, the hydrogen bonds between complementary chromosomal DNA strands will be broken. As a result, the two strands will be separated. Denaturation is necessary for hybridization to occur between complementary DNA sequences of chromosomal DNA and the fluorescent probe. This is followed by hybridization, in which the slide containing the sample is incubated with fluorescently labeled probe at 37 degrees centigrade overnight. During incubation, hybridization takes place between the fluorescent probe and the complementary sequence of the chromosomal DNA. After hybridization has taken place, the slide is washed in order to remove excess and unbound probes, which is necessary to reduce background signal. Following this, the slide is treated with a counter stain such as DAPI in order to visualize all DNA. Following this, the sample is examined under a fluorescence microscope in order to detect the location and number of signals. Similarly, by using multicolor fluorochromes, one can detect the presence of multiple target sequences on chromosomal DNA simultaneously. Fish is performed on metaphase or interphase cells. As already said, this technique is used to detect different types of chromosomal aberrations. For example, Samples were collected from both the parents and from the child. The target cells present in the sample were then fixed separately on 3 microscope slides and were then subjected to fluorescence in situ hybridization. Hybridization reveals the presence of fluorescent or positive signal on chromosome of both the parents, but the signal is missing from the chromosome of the child. This indicates that particular DNA sequences which are present on chromosomes of both the parents bind with the fluorescent probe. As a result, positive signal develop. But in case of child, due to the absence of the specific sequence, hybridization do not occur. As a result, fluorescent signal do not develop. This indicates deletion. Similarly, this technique can also be used to detect other aberrations such as translocation, amplification, and others. Friends, this is all about fluorescence in situ hybridization. For more updates, subscribe my YouTube channel, Sam Biology Class.